mean, you can see why we've had a lot of fun having Josh here the last few days. I mean, he's just fantastic. And it'd be great if he moved here, but, you know, until then, we'll continue to work with him in Sydney. But it's great now to have Carly come up and talk. Some of you knew Carly back when she was at Wayne State working with Mike Schei before he moved from Michigan to Iowa. But she was the first person that joined me out here. We actually both decided to start here before either of us moved to California. So it's great to have her come and talk a little bit more about what's going on in genetics. I think you can also see from Josh's talk that all of these things have to be integrated, which is the whole idea of what we're trying to do here. So it's not enough to just diagnose somebody, obviously. We have to help them deal with what they're dealing with now. So that's the management side of things, and you can see how futuristic that can become. And then we're using our understanding of the disability or issues with CMT and the genetics to come up with novel treatments. So I think that this is all part of the package that we're trying to develop. So with that, Carly is going to bring us up to date on some of the genetic testing.
So I wanted to take a few minutes and go through genetics in general, but I think that some of the more interesting things that are happening in the field right now is genetic testing and the way that it's changed, and some of the research that we've done surrounding genetic testing and the way that it's changed and what's happening right now. So it's kind of obligatory for me to go through what genetics are as part of a genetics and CMT talk. So anybody in this room that has not had the genetics and CMT talk? No, everyone's had it? All right, then I'm going to speed through this then. So this is a picture of chromosomes. We typically have 46 chromosomes in nearly every cell in the body. Chromosomes contain all of the information our body needs in order to grow and develop and function properly. They can be arranged based on their black and white stripes and their size into pairs, so that we have 23 pairs of chromosomes. We get one member of each pair from our mom and the other member of each pair from our dad. Likewise, when we have children, we pass along one member of the pair, and our partner passes along the other member. The first 22 pairs of chromosomes are exactly the same in males and females. They contain all the same information, all the same genes. The only place that we differ is in that last pair, which are the sex chromosomes. So females have 2 X chromosomes, and males have an X and a Y chromosome. Our chromosomes contain all of our genes. So we have 46 chromosomes, but 20 or 22,000 genes. So they're very small, arranged along the chromosome. And just like we have 2 of every chromosome, we have 2 of every gene, unless you have CMT1A, but that's a different story. So in this picture, the chromosome on the side is one's from mom and one from dad, and so then there's a gene, one from mom and one from dad. Genes are made up of DNA, which is essentially just a code. It's a series of letters that, when it's spelled correctly, lets the gene do its function. Problems happen if there's any change in this code, so like if a T was a C, or a couple were missing, or a couple were inserted in. Any change in the code could cause the gene to not work properly. In the past, we've called that a mutation. I think the field is getting a little bit away from that term. We're now calling changes in the code variants. And the variants have different terms. So there can be a benign variant, there can be a disease-causing variant, there can be a variant of uncertain significance, which many of you may have seen before. But so I'm trying to train myself to change my terminology also. So I'm going to try to use the word variant or change as I go through this, but I may slip back into mutation. In terms of the inheritance, there are 3 main forms of inheritance in CMT and then 1 minor form of inheritance in CMT. So the most common form is dominant inheritance. In dominant inheritance, all you need is to have a change in 1 copy of the gene in order to be affected. For a person who is affected, which in this picture is seen by having that red bar across, we'll say that's a gene, then in every pregnancy you pass down 1 copy of your chromosome, your partner passes down the other copy. So in every pregnancy, it's possible for the person on the left to pass down the copy of the gene without the change, and the unaffected parent to pass down the copy that doesn't have a change. So there's 4 possibilities in every pregnancy. That ends up being then that there's a 50-50 chance in each pregnancy of passing on the copy of the gene that has the disease-causing variant in it, and 50% chance of passing on the copy that doesn't. If a person inherits the copy of the gene with the disease-causing variant, then that person will have CMT, they will have symptoms. We can't necessarily predict what those symptoms will be, and then they also have that same 50-50 chance of passing it on in the future. If a person does not inherit that change, then they will not have CMT, will not have symptoms, and cannot pass it on. It just stops. In recessive inheritance, which is maybe about 5% of people in the US have a recessive form of CMT, you need to have a change in both copies of the gene in order to be affected. So on the bottom right-hand side here is the only person in this picture who's affected. It's a person who has a change in both copies of the gene that is a disease-causing variant. For a person who's affected with a recessive condition, the most common scenario is that both parents are carriers. Carrier means that they have one change in the gene that is disease-causing, but because they have a second copy of the gene that's working fine, they do not have any symptoms. Carriers don't know that they're carriers often until they have a child that's affected with the condition. So for them, the same thing with every pregnancy, they have a 50-50 chance of which one's passed on. So if both parents pass on the copy without the change in, then they have an unaffected non-carrier child. There's a 50% chance overall of passing on, one each passing on the copy with a disease-causing variant, so 50% chance of having a child that's a carrier, and then a 25% chance that each of them passes on a copy with the disease-causing variant, in which case the child is affected. However, for that child who's affected, the chance of them having a child that has CMT is completely dependent on their carrier, because a child that's affected is always going to pass on one copy of the gene that has the disease-causing variant. So if their partner has testing and isn't a carrier, isn't affected, then all of the kids will end up being carriers, like the affected person's parents, and not have any signs or symptoms or problems associated with CMT. If the affected partner is a carrier, then they end up having a 50-50 chance of having a child that also has CMT and a 50-50 chance of having a child that's a carrier. X-linked forms of CMT is probably about 12%, so this is the second most common inheritance pattern in CMT. And in this is the only time that it actually makes a difference if a person is a male or a female. So for a female who is affected with CMT, they have a 50-50 chance of which X chromosome they pass on. For men, when they pass down their X chromosome, they always have a daughter, because a daughter is going to get one X from mom, and then the only way that she can get her second X is to get it from dad. Every time dad passes on his Y chromosome, he's going to have a son. He's going to get one X chromosome from mom, and then the Y chromosome from dad. So mom can pass on either X to any of her kids. Dad's always going to pass on his X to his daughters and his Ys to his sons. So for a female that's affected with X-linked CMT, it's a 50-50 chance, no matter the sex of their child. But if it's a male that's affected with CMT, then he's only going to pass on his X to his daughters. And so all of his daughters will have CMT, and none of his sons will have CMT, because they'll have inherited the Y chromosome. Females affected with CMT1X are usually less affected than the men in the family, but they are affected, and this is kind of one of these things in the field that is still not widely accepted. But females have mild to moderate symptoms, and they definitely have CMT, and so it's really important to recognize the females in families with CMT1X. This is the minor form of inheritance that I talked about that's happening in CMT. So there's one mitochondrial gene that's been associated with having CMT. Mitochondria are the powerhouses of the cell. They make the energy. Parts of the body that require more energy have more mitochondria to power that. Mitochondria have a separate genome, so all of those chromosomes I showed you at the beginning, they don't have anything, mitochondria have their own separate genome, and it's circular, so it's not those nice lines. It's a circular genome. And we get all of our mitochondria from our moms. And that's if you think about sperm and egg, and that's the reason for it. So sperm need enough energy to get to an egg, and that's all the energy that they need. So there's like one mitochondria in a sperm cell. versus eggs they need enough energy to be able to grow a human. So basically, we get all of our mitochondria from that egg cell. The different mitochondria DNA may have different changes in them, so that if they have a disease-causing variant in one of their pieces in one of the genes, it's not necessarily in all of the mitochondria, as opposed to when we're talking about CMT in the rest of CMT, it's going to be in every cell that has DNA in it. So mitochondria, you're going to inherit all from your mom, but it might be from different stages of severity. So in this picture, the blue is affected. So here's a mom who's affected, but her kids can have inherited different amounts of that mitochondrial genome that have the disease-causing variant versus not having that disease-causing variant. So they can have different degrees of severity based on how much of that disease-causing variant they inherited. This is like the most complicated thing I talk about, I'm sorry. So you're gonna get if we see a mother that's affected, all of her kids will be affected to some degree. Versus if we see a man that's affected with mitochondrial form, none of his kids can be affected because he isn't really passing on mitochondrial DNA to his kids. So this is a very rare form of CMT. There's been like 1 or 2 families in the world identified with it. Getting into the more fun stuff would be starting talking about genetic testing. So when you go to the neurologist and you get a CMT type, you can get a CMT type without having genetic testing. And that's really based on the family history and the presentation nerve conduction studies. The only way to get a subtype, getting 1A, 2E, 4J, et cetera, is by having genetic testing and having positive answer. So you can get a CMT type. So type 1 is having dominant inheritance and having a demyelinating nerve conduction. I'm not going to talk about that now. I'm sorry. If anyone wants to talk about it, I can do it later. And then type 2 would be autosomal dominant axonal conductions. Type 4 is any type of recessive condition. There's type X, I failed to put on this, so any type of an X-linked CMT. You can have a distal hereditary motor neuropathy, which are predominantly motor symptoms without having the sensory symptoms. Hereditary sensory neuropathy, which is predominantly sensory without the motor symptoms, or intermediate conductions in CMT, and there's a whole slew of those that are coming to light. So if we decide we want to figure this out more specifically, we can go ahead and do genetic testing. Every letter corresponds with a specific gene and often the type of mutation. So that CMT1A is always a PMP22 duplication versus CMT1E is a PMP22 point mutation or a disease-causing variant within that letter code. And so we have found now, this is even outdated, but this is that picture of the chromosomes and has on the left side of all of these are genes that are associated with CMT, and on the right side are what they cause. So MFN2 causes CMT2A, PMP22 causes CMT1A, HNPP. So this is from 2014, and there's probably about 50 genes that are up on this, but you can even look around and see that there's some places that there is a type of CMT, but there isn't a corresponding gene that's been found. So as Josh was saying, we have about 84 that have been identified. This is from last year, and there's 50 on this picture. And we know that there's going to be a ton more that are found. So we still have a lot to do, and it's quite confusing. Oh, this is the picture of the mitochondria, that circle, the one little gene. So this is current genetic testing in the United States. In this, the way you can look at this is every other line is a gene, every other column is a gene, and then the next one are the labs that are performing that specific genetic testing. This is in the United States, there's 7 labs that are doing genetic testing for CMT. The code is on the bottom. And not every lab is doing every gene, but part of what I want to talk about is that a problem or is that good? The 4 that are highlighted here are the most common forms of CMT. So of people who have had positive genetic testing for CMT, 90 to 95% of them will have a change in one of these 4 genes. So there is testing for, there's something like 50 or 70 genes or something on this page. But of all of them, 4 genes are the ones that are really going to be the answer for most people. There are now large panels that can be done. So the smallest large panel is 17 genes, and the largest large panel is like 90 genes. Not all of them are specific to CMT. And so when I'm doing genetic testing, it's a question of which is the most, what's important for me and what do I think is important for the person sitting in front of me. And these large panels are really, compared to how it was in the past, much cheaper, much more efficient, time effective. But at the same time, as I was saying, not all of these genes are always associated with CMT, so it's a question of, is it even reasonable to be doing some of these really large panels? What happens if I get a variant of uncertain significance in a gene that has nothing to do with CMT? Then I have to explain that to you. And do you need to know this information even when it's not reasonable for your condition? So we did a study with one of the genetic counseling grad students here at Stanford, Allie Newhall, last year, and she was looking at what's the chance of actually getting a positive genetic testing result from these large panels. And Invitae, Athena, and GeneDx have very similar panels. Athena and GeneDx have published their paper, and then Allie got more information from Invitae about what they've done. And basically what was found that with these large panels, it's really only maybe a 20 to 35% chance of getting a positive result. And you can get a variant of uncertain significance. That's something that we don't know if it's disease-causing or not, or something that just makes you different from me, in 7 to 30% of those, in addition, 7 to 30% of cases. So then the question was, okay, well, we have 35 positive results. How many of those are those 4 common genes? And it ranges from 60 to 95% of the time it's going to come up as being positive one of those 4 genes, versus maybe 15% of the time total, 1 to 15% of the time total, are you going to get a change in one of these rarer genes that isn't those 4 common. So for me, it's a big question of, is that reasonable to do that additional testing? And so if you come to see me and you haven't had genetic testing, we're going to have a conversation about what's reasonable based on your presentation. So this is something that's evolving, and while it's cheaper and easier to do it, it's still, for me, a question about, is bigger better? So thank you very much.